Okay, so here is the cornea, and I thought I'd turn the light off over there for me. Here's your cornea, okay? I thought you might find it interesting, but it looks like microscopic. So this right here, this wavy pattern, it doesn't exist in a live cornea. That's artificial, because you have pressure in your eyeball. That's going to smooth that out, okay? So you have some epithelia on the inside and some epithelia on the outside of your cornea, creating, and here's high power of the inside, high power of the outside. So I thought maybe, I took these pictures so you might find them interesting to look at microscopically. I don't have any pictures of the lens, unfortunately. I have to take pictures yet. Okay. Anyway, you see in the visible wavelengths of energy. Light is energy. Okay? And the visible wavelengths are really narrow. Some animals can see in the infrared, like cats and snakes and other nocturnal beasts like owls, deer. They can see somewhat in the infrared. Lions. Okay? I mean, if you have a cat, you notice your cat loves to be in the dark. And you're walking in the dark, and your cat thinks you can see as well as they can. And what they do, they walk between your legs and you almost knock the cat outside. Cats do that. So these are the wavelengths that we can see in gamma rays, X-rays, ultraviolet, infrared, and short wave. Some people that have corneal surgery can see slightly into the infrared. Not everybody, but some people. And they don't know that they can. They just realize they can see better at night. Okay. That's all it is. Okay. They don't realize their vision has moved into the infrared. Not everybody can do that. So here's the layers of the retina. Okay. You have the ganglion cell layer, intersynaptic layer, bipolar layer, outer synaptic layer, and then the photoreceptor layer. And here's your pigment layer up here. Now, light has to travel through all this. And the receptors for photons are on these rods and cones. Okay? So light has to go through all this. And they newly discovered some new photoreceptors in the ganglion cell. These new photoreceptors. Okay? And they're the pigment, the epithelium in these photoreceptor areas, they're not derived from vitamin A. They're a whole different mechanism. The photoreceptivity that you see with is a derivative of vitamin A. Okay? We'll talk about that a little bit later. Right now. These photoreceptors are the same types of photoreceptors that cause the skin of a chameleon to change colors. This is in your eyes. So if you've ever noticed a blind person with a cane, they'll walk, they can't see, and they're going like this, and if you put an object in front of them that has never been there before, they won't even touch it with the cane, they'll walk around it, and then start walking again the pattern that they used to. They can't see it in the conscious level. But those photoreceptors, or newly discovered photoreceptors, are providing what we call an unconscious vision. A vision we can't bring to our consciousness. But it's there. Which is really weird. And humans can do that. And the blind can prove it. Now, the other thing that these photoreceptors do, if I had a light and I shined it in one eye, it would cause the pupil of the other eye to strip. Those are the photoreceptors. Those are the ones that cross over the chiasm. Okay? In addition, producing this unconscious vision. Is that a question?
Yeah, what part of the eye changes the image into the electric.
We're good. We're getting there.
Okay.
We're not there yet. Be patient.
Yes, sir.
Okay. So these are the layers, and I thought I'd take some pictures of this too. And we'll get to those images in a moment. Here is the optic center, the optic nerve. And if you have an ophthalmoscope, you look into the eye, and you follow the arteries and veins, follow them to the optic disc. The optic disc, it glows back at you. It's a really pretty sight to see. And again, if my ophthalmoscope is batteries charged, you could look at each other's eyes after class if you like. Okay? But you should look through the ophthalmoscope and see this disc. Hold on. So now you got what we call on the retina arterial venous crossing. Okay? So the arterial venous crossing, if it looks like this, high blood pressure. You can diagnose high blood pressure by looking at the retina. Okay? The venous crossing has to go all the way across where you have normal blood pressure. If you see these little yellow or white specks, those are microinfarcts in the retina.
Yeah, so that's
Diabetes mellitus. Diabetes mellitus. Those are microinfarcts as a result of uncontrolled diabetes. And that's a result of mobilizing too much fat and making atherosclerotic plaque and destroying capillary beds. She had a question.
I was just about to ask about the so the reason why the blue line was disconnected was because you had high blood pressure and it burst apart.
No, it didn't burst. The artery is pushing it, slamming it shut almost. Not quite. It's decreased. The artery is sitting on top. It's like the artery is choking the vein. It's not turning it off. It's not breaking it. It's still there. There's just pressure on it. Okay? Okay, so your blind spot is actually the optic disc. And your blind spot, my blind spots are right about here. Okay, in my visual field. Okay? And you can find your blind spot if you close one eye and you look at your fingernail. When your top of your finger disappears, then that's your blind spot. And it's a little, it's up in front of you. It's not on the side. It's right about, so if you look at it, you see that, and you keep looking forward. You don't look at your finger. My finger disappeared right there. Well, you're not doing it right. Your mind is lying to you. Your mind lies to you all the time. You have to really work at it to find it if you haven't ever done it. Because your mind is convincing you that it's not there. By filling in images for you. You do that all the time. You fill in images all the time.
I thought blind spots was like your peripheral vision.
Your blind spot is your optic disc where there's no rods and cones to see. That's an area of your retina that you do not have visual ability. That's why it's there. It's right about there. I thought it was further apart. Yeah. Okay, and everyone has one. You do have one. If you can't find it, it's because you don't know how. Your mind's lying to you. So here's your retina. I thought I'd take pictures of your retina. So this is the posterior chamber out here, and light has to go through this. This is your pigment epithelium. Here's your sclera out here. Here it is, a higher magnification, and I kind of like pointed out the areas that are according to this image that you saw a moment ago. The ganglion cell layer would be this layer right here. Let's see, this is the ganglion cell layer, and then this layer right here would be the next, that would be the horizontal or the bipolar layer, and this from here to here is your rods and cones. This is the inner synaptic area, this is the outer synaptic area. And here's the beginning of your pigment, right here. Okay. So the rods and cones are actually right here, and the cell bodies for the rods and cones are right here. So here's your cell bodies. Okay. Okay, so here's your rods and cones, your artistic rendition thereof. Your cones, they see what we call achromatic vision. They only see at night. Okay? And when you see at night, they only see black and white or different shades of gray. You're colorblind at night. You cannot see color at night. Okay? You think you can, but your brain is lying to you. If light is shining on an object, you will be able to see color. But if you remove that light from the object, it will become a shade of gray. Okay? And you can prove that to yourself by standing out here in this window and looking at cars pass. When they're under a street light, they'll have color. Then when they leave the street light, they'll turn to a shade of gray. But your brain will tell you what color it is anyway. Your brain will be lying to you. It will fill in the color for you. And a lot of times, color of a getaway car at night, the jury is told not to believe that you saw the color because you can't. You can't see color at night unless there's a street light shining on the object. Okay? And you see white light or bright light or color light with your cones. Okay? The rods respond to dim light used for peripheral vision, and the cones respond to bright light, have a high acuity color vision, and are concentrated in the foeva. You have a high concentration of cones in the foeva for color vision. So, what are the visual primary colors? And you'll be surprised what they are. The primary colors are not the pigment colors. Green is the primary color for vision. Green is the primary color, blue and red are primary colors. Now, pigment colors, how do you make green? Yellow and blue. Not for vision. Green is a primary color. Okay? But for pigment, it's yellow and blue. Yellow is a primary color. But in vision, it's not. Which is kind of weird. Okay? I told you vision's weird, and we haven't even gotten to the weird part yet. But so far, we've turned the image upside down and inside out. And now the primary colors are different. And light has to travel through all these things before it activates a receptor. And you have receptors for photons. Which is weird too, because what's a photon? We're not sure what a photon is. Some physicists call it a particle, some physicists call it a wave, and some physicists call it a particle in a wave. We don't know what a photon really is, but you have receptors for photons. Okay? Right? Mm-hmm. Okay. So if I were to have an apparatus and I made this room completely dark and I released one photon in this dark room, everybody would see it. And that in itself doesn't make sense. Well, maybe it does because light splits when it's going through a thin slit in an apparatus. It splits into three. So maybe like electrons, photons are everywhere at once. Who knows? But it's only one photon. Okay, so Photopigments undergo structural changes upon light absorption. The retina is the light absorbing part of all visual pigments. Retinal is, and that's derived from vitamin A. All photopigments involved vision contain glycoproteins called opsin and a derivative of vitamin A called retinal. Okay, so retinal is derivative from A. There are 4 different opsins. 4 different opsins. 1, achromatic vision, rods. 3 of them, color vision, the cones. Okay? So the cones photopigment absorb 3 different wavelengths, and those are the primary colors of vision, not the pigment colors of vision. Rods contain a single type of photopigment. Okay? So now we're going to start getting more weird. So here is a membrane potential at rest in a vertebrate for instigating vision. So you're at rest. So what happens when you see something? So this is dark. This is when there's dark, and then you add some light. You add light. You hyperpolarize. You don't depolarize when you see. Okay, so this is like backwards from everything I've told you before. Vision is weird. Okay, and there's a reason for this. So in this state, sodium enters the outer segment through channels that are steadily open. That's night vision, creating what is called the dark current. So the dark current is sodium current because it's maximal in the dark. The sodium-potassium pump maintains the current by maintaining this equilibrium of sodium and potassium across the membrane. So here's how it works. So here's the dark current, okay? The sodium-potassium pump is pumping sodium out and potassium in, and during the dark you have sodium current, meaning you are partially depolarized in the dark and you can't see. You're partially polarized. Okay? So in the light, when there's light, the sodium current stops. And as a result, potassium wins. And what do? You hyperpolarize. Then you can see. Now, the way this works is because of the neurotransmitter. Glutamate is a neurotransmitter. In the dark current is steady. In the dark, there is a steady release of an inhibitory neurotransmitter. A light-induced decrease in the dark current causes hyperpolarization. In other words, the membrane moves towards potassium's Nernst potential, and as a result, decreases the release of the inhibitory neurotransmitter. This is how hyperpolarization, rather than depolarization, stimulates us to see. So if I have an inhibitory neurotransmitter binding to its receptor on the postsynaptic membrane, it will inhibit an action potential in the dark. In the light, the neurotransmitter is removed, and then you fire an action potential because you remove the inhibition. You don't release the inhibitory transmitter. And then you see. I told you vision was weird. We're not even done yet. Okay? So here is retinal. It exists in the cis and the trans position. Okay? Here's the chemical Lewis structure of it. 11 cis, all trans. Okay. So what are we doing here? So light alters rhodopsin in the all trans position. And then there's an enzyme in your retina called isomerase that puts it back in this 11 cis position and combines it with opsin, so you have rhodopsin again. So when light hits it, retinal separates from opsin, and then you can see. when you take vitamin A, you can store it in the pigment epithelium as retinal or transretinal, and it can be used again when you need it. Now check this out. So, have you ever looked at the sun? Okay, and then you're kind of blind for a while? That's because you put all your rhodopsin in the transretinal and opsin position. You separated them, and now it's going to take time to put them back. That's why light can blind you. That's called photobleaching. I did that to my brother when we were little once. Okay. So it was pitch black downstairs in the basement, right? And I take him down there. I said, check this out. And it's pitch black. I said, what? And he can't see anything. I said, here it's my hands. I touched his body so he can. And I said, get closer. Put your head closer. And he gets closer. Check it out. And then I hit the button and it flashed. Right? And then we turned the lights on and he said, oh my God, I can't see. I'm blind.
Oh my God.
That's what he said. So we both started crying. I thought I blinded my brother. So it just took time. I photo-bleached all of his retinal. I removed all the retinal off of opsin, and it just took a little bit of time to put it back. And then he started seeing it again, okay? And then he attacked me, and we had a fight. And I had to kick his ass, too. Yes.
Okay, so like when it's snowy outside and like light reflects off like the snow and like when like some like play outside and then when they go inside everything looks all green.
Yeah, that's basically what we're talking. And some skiers get white out. Like you're out in the sun all day and all of a sudden you can't see. You can't see the moguls, you can't, everything looks flat. You've just whited out, you fatigued your eyes. And that can happen. That's also called snow blindness. And that's basically what happens. You photo-bleached. So vitamin A is an essential nutrient for your eyes. But if you OD vitamin A, let me tell you what's going to happen. Everything will start turning shades of yellow. You'll lose your color vision. Everything will look different shades of yellow, like shades of gray at night. And then, if you keep ODing on vitamin A, you're going to go blind. So, a little bit is okay, a lot of it is not good. Okay? Don't OD on vitamin A. So, here's how this system works, the retinal opsin system works. So, this integral membrane protein is opsin, and it binds retinal in transposition. Light hits it, and they separate, okay? The retinal comes off, okay? And then the opsin activates on proteins, a group of proteins called transducins. And transducins activate by phosphorylation phosphodiesterase. It's an enzyme that produces cyclic GMP instead of cyclic AMP. And cyclic GMP is removed, and when it's removed, you shut off the sodium current, and then you can see. So what does this remind you of? Huh? It should remind you of a G protein mediated signal transduction pathway involving G proteins. Guess what transducin is? G proteins. It's really a G protein signal transduction pathway involving cyclic GMP instead of cyclic AMP as the second messenger. Okay, so the pathway of light, so you see your fields, okay, and here's where it starts getting more weird. If I divided my eye in half, right, the nasal portion of this eye will cross over to the contralateral side. The temporal portion of this eye will stay on the same side. And that happens for both eyes. Okay? Crosses over at the optic chiasm. That's what crossing over means. We're crossing there. Okay, and then you go to the lateral geniculate nucleus of the thalamus, and things start getting scattered in terms of focal points. They're not organized. And then it's extended into the optic visual cortex, your optic lobe, occipital cortex, and it's even more scattered. Okay? So there's no real image that you can put together in your cortex. And then from there it goes everywhere else. Okay? So other optic tract fibers end in the superior colliculi. Okay, superior colliculus. Remember when you opened up the back of the sheep brain and you saw it looked like kind of like a butt-like structure? Superior colliculus. Okay. So some fibers go to the superior colliculus and pretectal nuclei involving pupillary reflexes. Optic radiations travel from the thalamus to the visual cortex eventually, so it ends up back there eventually. And then from there, just about everywhere else in the brain. Okay, depth perception, and we talked about this already, okay, so we don't need to really deal with anything here. So, read this.
Oh,
Yeah.
yeah.
You can all read it, right?
Yeah.
Oh, I thought you made it spelling.
What?
I thought there was a spelling mistake at the first one.
This is what a dyslexic's world is like. This is the way I read.
After some time, though, like if you're reading for a long time. Or
No, it's all the time. Anytime there's columns that I have to read, I flip them. Anything that's straight up and down gets... So if I have two letters next to each other that are straight up and down, I don't flip those letters. I flip the letters on both sides of those two columns. That's why I can't spell. I can spell as a result of memorization of big words. I can spell big words, but it's the little words I... So, that's what dyslexia is like.
the reason I can't know this